Okay, so this is what we are going to do today. We are going to look at the CCA analysis using this function provided by MATLAB in the statistics toolbox. And if you want, we can take a look of this so-called generalized eigenvalue problems, because this is the algorithm which behind CCA algorithm. Then we will look at this harmony, because we talked about the batch effects corrections. Harmony is one of the most cited or used method. People do this batch effects correction in single cell data. Okay, we will play with this result, this algorithm in set up in R and then run through the MATLAB. You will see how it works, and then you realize this algorithm will not change any gene expression values. So no change for your big X matrix. That's only change the location, the embedding of the cells. Then we will look at the MATLAB function for this dynamic time wrapping and DTW, and run its example and see what's going on. Then we will install Monaco 3. Monaco 3 developed in R, so we are going to set it up and then run through the MATLAB interface to call that function and see how you can achieve the pseudo-time analysis. So a lot of people using this, so we can try. Then I will show you, if you don't want to use any of those, because we have a dozens, maybe more than that, different pseudo-time analysis. But this is what I recommend. You just draw by your hand. You look at the embedding, and I recommend to use FATE. FATE give you the more dispersed, elongated embedding, and don't use t-SNE and UMAP, because you know this generative model will create cell islands, different clusters, but the relationship between them, there's no relationship basically. You can put subcluster here, subcluster there, or doesn't matter where, as long as they can separate. It's all driven by random number generators. So, but the FATE rely on some of this pairwise distance, between cells. So give you some good foundations to draw this from one place, move to another, to give you a more robust suggestion about the cell differentiation or development stage. Okay, so we will try that. At the very end, by the way, we will end the class this 15 minutes earlier, and today, okay. So I will show you, after you do this DEDV analysis, you get a list of genes. There are top selected genes. This mostly differentially expressed or mostly variably expressed genes. And then you can put into enricher, metascope, and also so-called gene agent analysis to understand the function of this gene set. I believe we already tried enricher analysis. But another one is metascope, and you can copy the genes, put into, and you can do the gene agent. It's a AI-driven annotation for the gene sets. But this is rely on a language model, so it's computationally heavy. So I'm not going to give you everybody a direct access, because you run this, you will get a list of genes. By click a button, you can send your results to gene agent. But because this is a very heavy duty computational platform, I don't want everybody to send that it will crash their server. So I gave you a password, which you can only have this password, you can use this function through the interface. Otherwise, there's no way for random people to use. Okay, I'll show you how to do this later on. Okay, so I encourage you to do this upgrading to try to upgrade your toolbox to the latest version, which is 2.7. Shouldn't have a great impact on what we are doing today, but there are some small improvement over the weekend. You know how to do this right now. You should from here, from add-ons, to uninstall your previous version, and then just reinstall the latest version automatically. You should have this, okay? On your computer, you also should have R installed. If you do here, you type R, you should either see RStudio or R itself. So that's what RStudio looks like. It's to update everything to make sure you have R installed, because there are a lot of inter-language communication tasks today. We need to set up this. If you don't know how to set up the link between this interface with R, there's video I just took to show you the button you click and how you found your R script. So if I play this, noise from the coffee shop. I just use a video. I saw it. So if you don't know how to do it, you can refer back to this, but I show you from this interface, we just do external, then R2, and then you select set up R environment. It should be point to a folder. This folder is where you install R exe file, executable file, and you just point to that, and this link should be set up. If this is not correct, you should try another, use another, you select the correct one. So that's what this R application or R script, they are the same. So you just click one of them. They should be set up. Any questions? I don't know on the Mac what's going on there. Yeah, if you can see, it's not active yet, but once we load some files, you can see the batch integration, the harmony in the R, and the pseudo time analysis, Monaco 3. So these two rely on the R environment, so we need to set that up. So this is the place. This is my R studio. So I want to, because we are setting up the R environment, let's just go straight to install everything we need. So that's a place I found from the Harmony website. And then this is a way to install Harmony. So if you copy this and put this into R, not into MATLAB, okay? So this is your R place. You put that in. Where R? Maybe I just don't care about the version. So that's what I did. I just copied that piece of code, and this will set up BIOS manager. And then from BIOS manager, you install Harmony. Yeah.
Where did you get that code from?
Yeah, so I just Googled for Harmony single cell integration. I found this page. I believe that's the authors. Don't worry if you cannot install it. To me, it's useless. But just let you know how to write, okay, if you need.
Can you please
paste the link? Okay. Yeah, so I can put in this. Here is link for Harmony. Yeah, so we should have a link to Monaco. So this is two links which you can install Monaco and Harmony. So Monaco will be taking some more effort than Harmony. Harmony is simple. So there's two links. So we are going to set them up. Okay. So that's after run the Harmony, you can try and see, use Harmony, and they should give you the message. Then you are okay. Okay, so this is a place we can also copy and paste. Oh, let's see. That will be hard. Yeah, so this monocle installation will be harder, and let me try. So if you do a monocle, it should have so on my system, there's no monocle installed, so I have to follow the installation and install those dependents first. So that's the place I copied. And this will take some time, I believe. Let's try. If I can do it. Yeah. And then the last step is to copy this. So I just follow instruction on that page and keep going and do this. If you come late, and that's what we are doing, just install Harmony and Monaco 3 in R environment. That's a link you have. Follow instruction. So let me know if you have this Monaco 3. Yeah. build R package. Yeah. So there are some issues because I'm using R 4.5. I need a so-called R tools. It will be very difficult for me. I need to reinstall this R2 according to the error message. So I need to do some extra work to make R2s available for the R version 4.5. So I have to download this according to the suggestion, and then after that, I should be able to compile the Monaco 3 and make it working in R. So I hope after I install R2s for 4.5, my RStudio should be able to locate where the newly installed R2s is located. It's located in the C drive. With that, I should be able to run this, okay, to install Monaco through the downloading the source code from GitHub and recompile on this machine. So I finished over there, and I hope this can work. Oh. Which step are you on? Are you doing this?
Reinstalling.
Okay. R. It's still it's R. Yeah, it's rely on a lot of packages, so it's had to recompile. So let's use this R tools. Okay, so I will just let this installation to run on its background. Here I want to show you the, so I'm going to simulate some cells. Okay, so by default I will simulate 3,000 genes in 5,000 cells. So this very simple simulation, the random number popping up some cells, random cells, follow some distribution, okay? Negative binomial, we talked about that. So I want to show you this is because we know those cells are homogeneous population, no structure. If we do embedding using, for example, T-SNE 3D, and it gave you one single cluster, okay, as expected. And it's round in this sphere, this ball, and indicate that no structure, this homogeneous population, this purely random cells. If we do this using existing node, if we do UMAP, either 2D or 3D, so I try this random simulated cells. So this is UMAP result. Okay. Let me select. If you do UMAP 3D, that's what you get. This is not a perfect sphere, and you can see the elongated, the same. There's one end, another end. But we know this is not a real, maybe just some random shift of this profile of the cells. Now, if we do FATE 3D or 2D, I will run both of them. So this is something you can test because you know the answer. Your simulation, there's no structure. A single, highly unified population, homogeneous. But the different embedding results may look very different. FATE will introduce some, how to say, exaggerated structure. Even though you know there's no structure, FATE will give you some structure. Okay. Okay, so Monaco is installed correctly on my, oh, it's not, still not. So let me keep doing this. So this is the result for FATE. Okay. Well, although we know there's no structure, FATE gave you something out. It's hard to explain, but this is an ability which FATE can we can utilize this. It could be no structure, but still there are very small small step difference. But when you apply to real data, when real structure is there, or the pseudo time or development stage in the real data, maybe fit is sensitive enough to capture this. Okay, that's what we hoped. So come back to the Monaco. I installed it, but it's still not there. So if I try to load this, no package called Monaco. Oh, this is GRR not installed. So maybe I should install GRR. I don't know. So I decided to give up. There's too many dependencies. I don't know, there's comparability issues. I don't know how to install GRR in R package. Well, if you can make it successfully on your machine, you should be able to run. Let me see if I can have this on my laptop. It's really version dependent and machine dependent. Yeah, it seems there's some issues with this package, so it has been removed and deleted from the archive. Yeah, so I don't know how to solve this problem in R. Looks like their essential dependent library has been removed from the archive, so you cannot install GRR, and then so the whole Monaco 3 package we cannot install.
I'm getting the same issue.
Same issue as this.
Same issue.
Yeah. So that means in this moment, shouldn't have anyone is using Monaco 3 because you cannot install it.
And over there in the error message, it's written a version of this package for your version of R might be available elsewhere.
This place?
I mean, it's written version of R, so you're using 4.5.2. I'm using 4.5.3, but I'm still getting the same
error, yeah. The message is like this. It has been removed out from. So that's the announcement from the place. So last year, some issues with the package.
So that means nobody can use Monaco.
Yes, so if someone claims they are using it, I highly doubt it what they are doing.
Once you install GR, it works.
Yeah, so if you can, suppose. Have you, you can do it? Can you?
Yeah.
Oh, you did? Okay.
I don't know if I can. When you do install packages, you put the link to the GR tab file, then you install. Then after you run the Monaco install, then it works.
Oh, so what should I do here? So I should restart
I don't
my
know if I can put something in the
chat. Okay.
posted something in the channel.
Mm-hmm. So you just try install the older version, something like that, from a different place. Yeah.
And after this, then you run the Monaco install of the.
Yeah.
I tried opening the link and it says object not found.
You have to, what I posted, you have to post
Copy.
it in R.
You copy this, put in R. Yeah. The whole thing. That's good. Yeah. So now, yes, it's working. Thank you, Felix. And you're the hero here. And so, indeed, after this library installed, just miss that. I don't know how you figured out. Yeah. Okay. It's good. So all you need to do is just copy this, so we can install GRR, a particular version from from an archive, basically. It's before it has been removed out, then we still keep using it for some of the function. Hopefully, we okay. Okay, so we spend a lot of time on this and we are ready to move forward. Yeah, since we freshly installed this R package, so we're going to call these two packages through the interface because we are ready to use them. So we just load this example data and then look at the batch ID. Okay. So these two batches are separated. So let's try Monaco to merge them. Mm-hmm. Yeah, so this is what Monaco did. It merged the cells. It was separated and merged. Okay, I hope you can try on your own. And Monaco is iterative. You can run this after you get the merged. You can run one more time, and then it will merge closer even, okay? Until you're happy with the result. Let me know if you can run it. So, but that's what Monaco does. So I run one more time. Okay, so that's the result of merging. I can run another to show you before and after merging. So I have this side by side, colored by batch ID, and this is before merging. This is after. Okay, I have done my job, and I showed you how to do this harmony batch correction. This is the before. Okay, before merging. This is after. Run 2 rounds of the Monaco, not Monaco, harmony. That's what I achieved. So I just will go around and make sure you are okay with this, and we can have a break. So what I just did is just use example data. So you load your data in and show the batch using color label, and then run Monaco. Then the cells start to merge according to the batch ID and the. Let's check whether I want to know whether the same cell type will be merged together. Okay. Maybe that's the point. So if I look at this merged version and then to have a multi-grouping the cell type at the same time. So this part is merged. Looks like they all alpha cells. But these two parts are not so they merged as well. But they are different cell type. Oh, not merged. It's good. So okay, so I show you what I'm doing here. Through comparison of this before and after merging, what I found is most obvious result of this batch correction is to merge cell from two different batches into same group with here. Okay, so this place is where merging happens most. It would make sense if the merged cell belong to the same cell type. So I come back to check whether this merged part here is the same cell type. Yes, so they are these alpha cells. So the alpha cells have been separated into two batches before the batch correction. But now after harmony they merged. So maybe it's good. So if I do before merging, I have alpha cells have been separated into two clusters. That's right. So this dark navy blue are alpha cells before merging, right? So you have two separated. They are same cell type but a different development stage. 8 weeks, 14 weeks. But harmony will merge them according to the results here. So this merging to me makes sense because they are same cell type at the end. I don't know if you can do similar thing on your machine, but that's what I can demonstrate using this pre-processed example data. So remember, our strategy was to separate every cells into many small clusters, and then annotate them. If they are same cell type, they will be colored in the same way. Monoclonal maybe take a different strategy. They try to push cells into the same cluster before they do this annotation as a whole group. Yeah. Okay. Any questions? Okay, so 5 minutes break. Okay, so we are going to use this example to run Monaco 3 and see whether we can get some luck. By the way, there are multiple cell types. I think we should clean it up to see, okay, we need study the differentiation in alpha cell only, maybe. So I'm going to extract alpha cells. Because if you have multiple different cell types, how the Monaco will run, and but if you have a stem cell and something derived from stem cell, you put them together, makes sense. But here, they all differentiate the cell type already. So I think our question is within the alpha cell from week 8 to week 14, whether there's a trajectory we can follow. So I'm going to extract only alpha cell and clean the data and send it to Monaco and to get some results and see whether we can do anything with it. So in order to extract alpha cell, and let's try select cell by class. Okay, then cell type. So if you don't have this example, you can load the example data. So let's do alphabet. So alpha cell, I want to look at the size as well. So we have a lot of alpha cells in this example. That's good. So select them. So I close the original, so we have this many of cells. We need to do a QC, remove some genes with too low expression values. So this change our gene number, only remove 80 genes, so it's okay. So now we have this alpha cell in 2 different time points. We are going to make a trajectory using Monaco 3. And the Monaco has its own embedding method, so we don't have to do our own embedding. So just click this and let it do, or ask to select the root cell. So we need to, that's one of the input required by Monaco 3. So we need to brush a cell as a root. So we believe this could be a root here. Okay, brush. Select it, continue. So this program will send our cells and expression, including which cell you believe is a root, and to the monocle. Monocle is processing its own embedding and build the falling. I don't know. Yeah. It could be a problem, and I will, so we cannot run this for some reason. Do have a bug in the code. So I will try on my laptop, see if I can achieve anything. If not, I just have to go back and figure out what's going on. In which step we have this error message. We'll try with all the cells. Yeah, so the same problem. So I do have a bug in the middle of some, yeah. So I will figure it out, but in general, you will get a new trajectory and all assigned pseudo time points, okay? So I will figure it out and give a new version next week, and we can try make sure they're correct. So now, using this example, let's try the one I suggest. We should do this manually. I'm going to use this. Okay, we are doing. Let's see if I can make it. We are going to do a new embedding. No, we'll do everything here, okay? Because I extracted the alpha cell, and it was in with another cell. Now, I just redo this and see whether okay, so this is the result I got from all the embeddings, including 2D, 3D. It's clearly, and there are two separate clusters. They are different stage of development. So we cannot draw anything from this end to that because there's no trajectory or pathway. But fate seems give you some information about trajectory. So we are going to use fate 2 to draw this trajectory. We start from this end and move to that end, okay? So on this page, let me close the other one. We need to use 2D because this drawing needs 2D, so we found 2D fate. And then from plot, we see a cell trajectory. Okay. So this is a suggestion that encourage you to use fate embedding in order to do this. So you click yes. If you don't want to draw, you can have automatically generated. So the spline fate will give you a result like this. It's not ideal, so we try. Sometimes give you really good results if you have a clear trajectory, so you don't have to draw it yourself. Another way, if you do remove, if you do the principal curve, just automatically draw a curve for you. This is not very good, especially give you the sparsity of those cells. So those algorithms not help. So we need a manual drawing. Okay. So trajectory. See, yes, and remove existing. So we are going to do this manual drawing. So draw a curve, and we will start from this end by click. Okay, keep clicking. At the end, double click maybe. Let me see. It should be right click. I don't know how to stop. Return key. Then you stop. That's the draw of your trajectory. So after you click finished, you just keep the key return, and then what you draw just put in there, and you can save it for later use. Then you can find some genes which. So this is a pseudo-time analysis by hand. Okay, you draw. You believe the cell trajectories go that direction, and after you create this trajectory yourself, you can see, okay, this is time 0, this time 1, time 2, and give me some genes which follow the trajectory. I can have some picture like that. And this curve will be saved in temporary files. If you want to reload them back, you can. Okay, so that's the way we do things. Do you have some doubt? It's not a data driving, not an algorithm driving, it's by hand. But sometimes I believe this is the best way, because you see the cell, the change. You have other information of marker genes. If you show gene expression level, project on some of the cells, and you have your biology knowledge. So I would believe this rather than any algorithm itself. And clearly there are a branch here. So if you do a different curve, so you go from here to there, and you have found some other genes which follow the trajectory in some correlation with the time. Okay.
The cutoff for like the expression that if it's like the lower ones, like the one around 0 to 1, do you consider them with more confidence as the ones that have higher expression?
It's so if you have a function at a time, so time monotonically increased, 0, 1, 1.5, 2, and then you just do the regression of the gene expression with the time. And some maybe have a correlation 0.7, some 0.5, 0.65. So you can have a different cutoff. But this just gave you some.
My concern is that since the y-axis expression is there, can you attach some significance to how reliable the pseudo time is based on the expression? Like the ones that is the expressions like around 3 versus the ones that are low.
This is a really high expression. Yeah.
Is there any way, or it just take it as it is?
I just take it that it is, and if you interpretations, I don't know which gene this. This should be insulin 1. This highly expressed in the alpha cell. But if the value is high but not follow the time, there's no variation, then you don't detect them. But this based on your regression. So if you consider average expression as a factor to do the regression, or you only consider by the expression level, by the rank and in the time. So you can have different ways to do this feature selection to find the genes which help you to explain this time increase. So once you have T, so every cell have a different T. So you have T0, T0.01, T0.121. So every cell will have its own unique, because we project cells onto this trajectory. So every cell will have its own time point. If you have that, you can do a lot of feature selection. So you know the lasso, rigid regression. So we do. So once you have time, then there are feature selections. So the curve I just showed you is using Spearman correlation. So some very low hanging fruit genes which follow the change of the time. But if you are interested in some genes which was low, but in the middle they're high, then come back to low, some nonlinear, you can use lasso or even this QUBO-based feature selection because you have a T. Once you have a T, you can do a lot of this time theory related analysis. Okay. Let's see if I can export the T to. So if I do this, select dependent variable y. So I do have so once you finish this pseudo time analysis, either by hand or if we can successfully run Monaco, you will have a variable. So if you click here, you have a pseudo time. Okay. You have a pseudo time. If you click, then it gave you continuous the current figure. So you can have the pseudo time color for different cells. With that, because you have T, you can do feature selection. So this helps you to find the gene which help you to explain your pseudo time. So you select this pseudo time as your variable you want to explain. Then you can have, I will do the transformation. Yes, do a transformation. One of the transformations, so you will see the lasso analysis. So this will help you to find the feature genes rather than just rely on this correlation. Okay, this will give you a table. So which gene combinations will give you the best results? Something like that. Okay, so it helps you to find the top genes when they are combined to give you an explanation for the time. you probably need to revisit Lasso, the one of the lecture we have regression and regularization, how you interpret these results. But you do have a list of genes, okay, coefficient by. Those genes explain the time selected top ones. Okay. Okay, so for the rest couple minutes review what's on our agenda. So we did this. I think I can quickly show you how to do this, okay? So let's copy this key. So you need to copy this. Come back to this. You see, you can load example data here. Yeah, you see this is not accessible for you for the gene agent analysis. By giving the reason I just told you, because it's very computationally heavy, I don't want to create their working load unnecessarily, so I restrict this. So there's a lock here, okay. To unlock it, you have to go help and then put your validation key, and here you give that particular key, and then you click OK. Then you should be able to use this. It's unlocked. Okay. The input is very simple. Just a list of genes, and this list of genes will send it to that server to run. Okay, so this is the key, and then you can unlock this. The input will be the same as an enricher. Enricher will also get a list of genes. So if you look at the enricher, so this is like example genes, okay? Then you send this to, if we don't need a background, then no. And if you want to use web-based, this is just a web page. Your genes is here, and then send it to enricher. You get the results and what those genes, 200 example genes, are doing, the function terms. Okay? So same thing if you do enricher, this gene list, every time is a random set. Okay? Just for demonstration purpose, you can run the no background API based, then you will select which data set you will search against it to. Okay, those are the commonly used. There are a lot of others. So you will search your database, your gene set against those database. If you see done, and then you have the table. So another bug. So you have table. You should be able to save for your report. Okay, so I will figure out these errors. Same thing here, you need a list of gene to do in gene. So I don't have a long list gene. So this is just an example. So you send this to this gene agent. Yes, take some time because this is a place the gene agent will help you to analyze the gene set you gave to it. And you will see this output from a language model. Just start to search the literature's database. So all you have to do is just get your results using DVDE and get a list of genes and paste in that table, and it should be. You can certainly directly use this web interface, but sometimes you forgot where the link is, but you can through the MATLAB interface to achieve the goal. So you have to wait. It will generate a report for very thorough analysis of the function of those set of genes. It's very powerful and give you the large language model enhanced gene enrichment analysis.
The gene set that I selected, it does not have any biological relevance.
That's really cool, and it's hard to find any gene set without any function. Yeah, looks like this error message was from the server itself. So maybe not a bug in my code. It's just this particular service, cloud service, not available at this moment. Maybe. Yeah, so now everything is done, and you can have retrieve all these results here. You can come back here as well. It's done. Yeah. So you should have a better results. Let's see. Okay, something wrong here. Yeah, so I do have a code which rely on MATLAB reporter generator to read this information back to generate a Word document. But you don't really need that part because it's already very thorough and give you the report for every step. Okay? Yeah, so definitely this is a place I needed to be prepared. If you don't have that reporter generator, should warning you rather than just give error message. So it's something we maybe next version I will fix those small problems. But that's the whole idea about once you have DVD generated results, you should be able to use different tools to understand your results. I believe at the end, I just show you the dynamic time routing algorithm. Then we are done with today. So there's a function we can use. I'm going to just copy the example they used to do this DTW. So the example is you have X and Y, two signals. Then we copy this at a different frequency. So you get X and Y. Oh, I didn't have that. So yeah, so that's in the signal processing toolbox. Okay. Yeah, so this function is inside here. So I don't have this, but if you have on your machine, you can try. I gave you a notation here, so we need this. Okay. We can try this. CCA, okay. So this is in the statistics toolbox. I definitely have this installed. So what we need to do is look at this documentation and run examples. You have two matrices, X and Y. Then you will rotate them separately, but the achieved result is after rotation, the correlation between this embedding will be the maximized. So that's what our X and Y data from. Let's copy this. So it's big car data. And then we remove some data point contains missing values. Okay, the first matrix X contains 1, 2, 3, the first three variables. So that's three variables define each car. And then your Y axis, your Y matrix contains 4 and 5, another 2. So they sampled from same cell, same cars, but measured in different variable combinations. Now, we need to find a rotation for X matrix and a rotation for Y matrix to align them. Okay? So this is a CCA. To do this, R is the result which achieved, believe. Okay, two correlation coefficient. For the both of them have been reduced dimension to 2, okay? So if you look at the dimension, the first matrix is 3, okay? Depends on how many cars you have. The 406 cars, then 3 variables. And the y is 2D, 406 and 2 variables. And then you rotate and embed them into A and B. They're 2D, and then you do the correlation. And for the first dimension correlation, 0.8, second 0.6. So let's look at A, the dimensionality A. This is rotation matrix maybe. And the B, okay? So A and B are the rotation matrix. And the UV should be rotated. Yeah. So what's X? Yeah, so X is 392. I believe the original data have more than 400 cars, but some cars have missing values have been removed, filtered out. And your XY is 392 and reduced dimension to 2. So that's the CCA. That's correlation, okay? Okay, so that's all for this week. Let me know if you have any quick questions. I will stop here and give back your 15 minutes before noon. Anything else missing? Everything? Okay, wait my next release of the toolbox. I feel fix those small bugs, and then you should have a more robust tools to use. And I showed you how to do this cross-language calling R from there, install R. Sometimes it's frustrated, and hope Flex, you are very good today and help us to install this. We should document this, and so other people know how to make it run. Okay, I hope the rain is stopped. So don't tell other people this key, okay? Keep it a secret. Okay.